Hi, I'm Mustafa Sahin from Children's Hospital. I'm a child neurologist, and my lab works on tuberous sclerosis. It's my honor to introduce the next speaker, Dr. Vijaya Ramesh from Mass General Hospital. We're going to shift gears and go from finding genes and asking the question, how finding genes improve the lives of patients with autism. And many of us in this room believe that one plausible approach is to take advantage of single gene disorders that have high penetrance for autism, such as Fragile X, tuberous sclerosis, or Rett syndrome. And as Chris alluded to, there are currently clinical trials in each of these diseases that are ongoing in this town right now. And this is where Vijaya's work comes in. Without the basic science studies that are done in many labs around the country, including Vijaya's, we couldn't have gotten to where we are right now. So for that perspective, I want to just highlight two of our major contributions to the tuberous sclerosis field. In about 10 years ago, Vijaya's lab was the first to show that the TSC1 and TSC2 proteins were found in the growth cones, which are the tips of these cables by which nerve cells connect to each other. That work, or that finding, has led to work in many labs, including mine, to try to understand how TSC genes contribute to brain wiring and how brain wiring can go awry in the brains of patients with tuberous sclerosis and other forms of autism. And her second important contribution was a collaborative study with Alcino Silva and Dan Enninger at UCLA, where they showed that there were learning problems in mouse models of tuberous sclerosis. And these learning disabilities, learning problems, could be treated with a very short course of a drug called rapamycin. And with this treatment, the mouse learned essentially as the control mice did. So that particular study led directly to our clinical trial in children with tuberous sclerosis right now that's ongoing across the street in Children's Hospital, where we're looking at neurocognition and autism and see if we can treat these deficits using drugs in the same family as rapamycin. So with that, I'd like to introduce Vijaya. She's going to talk today about a new cellular biological finding that she's made, an interaction between a large molecule PAM and the TSC pathway.
Thank you, Mustafa, for the kind introduction and giving a brief our contribution to the TSC research. And thanks for the Autism Consortium for giving me an opportunity to speak here. So it's going to be switching from genetics to more functional stuff, and I'm going to be talking about this large protein called PAM, which functions as an E3 ubiquitin ligase. Just to tell a brief background about TSC, it's a multi-system disease with a frequency of 1 in 6,000 to 1 in 10,000. And one of the most common neurocutaneous disease caused by mutations in TSC1 or TSC2 genes encoded by protein known as hamartin and tuberin. And patients are characterized by slow-growing benign tumors called hamartomas in many organs, including the brain. And the CNS defects include seizures, mental retardation, and autism. Anywhere from 25 to 50% of the TSC patients show autism spectrum disorders. So it's one of the classical syndromic disease with autism. So the pathway is very well known. Many people, Drosophila as well as in the cell biology field, people many years ago, quite a few years ago, placed TSC proteins in this pathway. In this MTOR signaling pathway. TSC1 and 2 function very tightly together as a complex to negatively regulate the signaling molecule called MTORC1. When TSC proteins are mutated, this protein is abnormally activated, resulting further activation of these molecules S6K and S6, which are detected by phosphorylation using specific antibodies. In addition to mutations in TSC genes, upstream signaling, either through PI3 kinase AKT pathway or the MAP kinase pathway, can also lead to inactivation through phosphorylation, leading to the same results, hyperactivation of this pathway. This is primarily in growth syndromes, in tumor-associated diseases like TSC, NF1, as well as another tumor suppressor P10, also another tumor suppressor called LKB. So it's kind of interesting. Many tumor suppressor genes are placed in the pathway, and my lab recently also placed the tumor suppressor NF2 in this pathway. So in the Drosophila, you can see when this pathway is activated, you have cell growth, which is cell getting larger, not only proliferation, but cell getting larger. In the Drosophila, you can see the eye size getting larger when this TSC genes are mutated. And also you can remove the protein, the TSC genes in the mouse brain, you can see the macrocephaly or enlarged size brain, which is all related to growth. So what happens in the brain itself when these proteins are gone? In actually either through synaptic activity or neurotropic factors. Many of the work has come from Mustafa's lab recently, nicely showing when these synaptic activity or the neurotropic factors are activated, TSC proteins, through the upstream signaling here through the MAP kinase pathway or the PA3 kinase pathway can inactivate, resulting in the abnormal activation of this pathway, which is very critical for synaptic protein synthesis. So this mTOR signaling plays a crucial role in synaptic protein synthesis, which is, again, necessary for synaptic plasticity, learning, and memory. So what I want you to remember is it's not only the hyperactivation, you cannot have the underactivation or lower activation of this pathway either. You need a balance of this pathway. You cannot have hyperactivation of MTORC1 or lower hypoactivation of the MTORC1, which will all affect the synaptic protein synthesis. Few years ago, in 2004, when we were working on TSC proteins, we identified this huge protein with 4,000 amino acids that came out of originally from Mike Bishop's lab as protein associated with c-Myc. So that's why the name came PAM. So we just retained the name. So we found the extreme C-terminus to be present in a complex with TSC proteins. This is an interesting molecule with several motifs, and we are still trying to understand, largely because many labs don't want to work on this protein because it's so huge, a 500 kilodalton, and seeing it on a gel, it's not easy. So it has a guanine exchange factor activity or CC homology domain at the N-terminus, which we still don't know whether it is a GEF for any of these small GTPases. There's a MYC binding domain, which is c-Myc, which also plays a role in neurons. And then the C-terminus, the ring zinc finger, is a hallmark for E3 ubiquitin ligase activity. So E3 ligases are important. Angelman syndrome is a E3 ubiquitin ligase, which is mutated. And E3 ligases are important in brain to keep the synaptic proteins in balance. So the other interesting thing is this protein is highly conserved in Drosophila, C. elegans worm, and zebrafish, and they all function to regulate axon and synapse development. So this is a key protein that is involved in functions as a synaptic growth regulator, functions in axon guidance, outgrowth, and synapse development. And again, it has a C-terminal ring zinc finger domain, which we identified as a TSC interactor. So my lab showed originally when we identified that through different, both in Drosophila and mammalian system, it could be functioning to ubiquitinate TSC and destabilize this complex. This is one of the hypotheses or the results we're leading to suggest that in addition to phosphorylation, TSC complex could be ubiquitinated and regulated, degraded by this large E3 ubiquitin ligase. So we went on to address that question. If it is an E3 ligase, it has to bind to E2 enzymes. So ubiquitination, any cell has 3 enzymes, E1, E2, and E3. First, the ubiquitin is added to E1, which is then transferred to E2, which then subsequently binds to E3 and transfers the ubiquitin to a particular substrate or a molecule that is marked for degradation. This is how when the ubiquitin is transferred to the substrate, that particular protein is degraded. So if it is really PAM is an E3 ubiquitin ligase, it has to function, it has to interact with E2, bind to E2 enzymes. So we asked the question, is it binding to E2 enzymes? And it was binding to more than one E2 enzymes, actually 3 different E2 enzymes, strongly suggesting it is functioning as an E3 ubiquitin ligase. We also showed it can in fact ubiquitinate TSC2, and when TSC1 protein hamartin is present, it prevents the TSC2 ubiquitination. And some of the patient-derived mutations from TSC, when it can no longer bind to TSC1, can be easily ubiquitinated by this protein. We undertook some experiments. At that time, we didn't have knockout mice for this. So we decided to do RNAi by artificially removing this protein in neurons. It's highly expressed. One of the easiest places that we can see this protein is in neurons, in brain. So when we do RNAi, we ask the question, when this protein is gone, it can no longer ubiquitinate TSC2. Can the TSC protein could be stabilized, and MTORC1 could be downregulated. So this is actually when MTORC1 is downregulated, the readout molecule is phosphorylation of S6K or S6. And that's what I'm showing you here. When PAM is reduced by RNAi, TSC proteins are stabilized. Both the phospho S6K and phospho S6 were reduced, suggesting, in fact, mTOR signaling could be downregulated. This is interesting. As I told you, you cannot have hyperactivation or hypoactivation, which is going to affect the synaptic protein balance there. So all the work that I'm going, so this is to suggest that in addition to phosphorylation, TSC proteins could be ubiquitinated, and PAM could be an E3, important E3 ubiquitin ligase regulating this pathway mTOR signaling. So which could affect the local protein synthesis in the synapses as well as synaptic plasticity. So we have now moved on, and all I'm presenting is almost going to be submitted as a manuscript and published work. So we, two different groups had knockout mice for this large protein, Aaron D'Antonio's lab at WashU as well as Sampaf's lab. At Stanford, they had Salk Institute, they had the ENU-induced mutagenesis. However, the mutations were very different. This PAM has 85 exons from Aaron D'Antonio's lab. By some mechanism, somehow they removed in-frame deletion of exon 8 and 9. This is actually a knockout, genetically engineered knockout, but they didn't realize when they were making the construct, but it turned out it was in-frame deletion of exon 8 and 9. However, the ENU-induced mutagenesis from Sampaf's lab came with a stop codon in exon 63, removing the extreme C-terminus of the protein. So we thought these are two interesting, not completely null, but it could give us some functional aspects. So for both models, the heterozygous mice appear normal, fertile, whereas the homozygotes die at birth due to, again, innervation synaptic defects. They cannot take breath when the mice are born. The neuronal defects observed in mice suggest this PAM, which is the mouse homolog, called PHR1. It's required for axon guidance, outgrowth, and synapse development, so which is consistent with the C. elegans in the Drosophila. This is a very critical protein for synapse development. And again, both models are not true null because it's such a large protein. So we kind of thought it'll be interesting to ask the question, what happens to mTOR signaling in these two models? We kind of predicted the C-terminal deletion with the E3 ligase gone will have reduced mTOR, whereas the N-terminal 8 and 9 in-frame deletion will not have a defect in mTOR. To our surprise, actually, this is, I'm not going to go into the blot results. So this is reading out for phospho S6K and phospho S6. And when we saw the N-terminal 8 and 9 deletion, in fact, it was more strongly reduced for mTOR signaling than the C-terminal E3 ligase deletion. This was kind of very surprising and kind of, intriguing finding for us that mTOR being affected. So then we asked the question, what happens to TSC proteins? If the C-terminal deletion E3 ligase is gone, the TSC proteins need to be stabilized. That happens in the Magellan mice, which has the C-terminal deletion. However, in the N-terminal mutation, the TSC proteins are intact. So this suggested to us that the C-terminal deletion is as expected, functioning as an E3 ligase through TSC proteins controlling mTOR signaling. However, the 8 and 9 deletion is still involved in mTOR signaling independent of TSC proteins. And DLK is actually a molecule in P38 MAP kinase signaling that is the substrate for PAM homologs in high wire and C. elegans, which are the Drosophila and C. elegans. DLK is a substrate. However, we show that in mammalian system, DLK is not a substrate. It could be other targets instead of DLK. So we took one step further and asked what happens to neurons from these mice when you make the primary neuron cultures and treat with BDNF. So when you treat with BDNF, a neurotropic factor, both of these pathways are activated, resulting in TSC2 inactivation, further activation of phospho S6. So when we take the neurons from Magellan mice, we do see the activation in the presence of BDNF, the phospho S6 here, compared to the wild-type controls. However, when you see the 8 and 9 deletion, you do not see that, suggesting again, in the N-terminal deletion mice, they are not functioning through TS, they are bypassing TS and functioning either at the level of Reb or MTORC1, which we really do not know. So this is again highlighting that this huge molecule controlling is controlling MTORC1 signaling, one through TSC, one independent of TSC through two different domains, which is kind of not surprising having this such a big 85 exon protein. So then we ask the opposite question, okay, when you remove this protein, you are hypoactivating, mTOR signaling is downregulated. What happens when you overexpress this protein, particularly the N-terminus and the C-terminus? So what I'm going to show you here, when you either overexpress the N-terminus of the protein or the C-terminus of the protein, the MTORC1 signaling is strongly activated, regardless of whether you express the N-terminus or C-terminus. Again, suggesting PAM is an important regulator of MTORC1 signaling, and overexpression of either the N-terminus or C-terminus is activating, and 2 independent domains are regulating MTORC1 signaling. So the hypothesis we came was PAM regulates MTORC1 signaling through functionally 2 independent domains, probably the E3 ligase domain at the C-terminus and either a putative GEF domain or unknown mechanism at the N-terminus. And we also hypothesize part of the neurodevelopmental defects that you see in these mice may be explained by abnormal mTORC1 signaling, particularly because of the synaptic protein synthesis. So we decided to cross these 2 mice to make compound heterozygous. So if you take this 8 and 9 deletion mice and cross with the Magellan mice, you're going to end up with one allele with 8 and 9 deletion, the other allele with the C-terminal deletion. So if they are truly functioning through independent domains, according to genetics, they should complement each other and rescue the mTORC1 signaling. If they're not functioning through 2 independent domains, this may not happen. So that's the cross that we undertook, and interestingly, mTORC signaling was rescued. In vivo, suggesting indeed these 2 domains are independently regulating. So we do not see in the compound heterozygous. I'm showing you representative brain results, Western blot results here. Many of the, at least 15 or so compound heterozygous mice we tested do not show any longer downregulation of mTORC1 signaling. Then the question, so this is to sum up here what I have shown you with the compound heterozygous. So when you take one allele of 8 and 9 deletion, one allele of the C-terminal deletion, they still, however, die at birth, indicating that one entire wild-type copy is required for these mice to survive. So homozygous die at birth, and also the compound heterozygous die at birth. So you at least need one functional wild-type copy. However, mTORC1 signaling is not attenuated, indicating that mTORC1 downregulation is not responsible for lethality. Interestingly and importantly, we show that intact N-terminus can complement the C-terminus, so their functional complementation, so that there is 2 different domains regulating mTORC1 signaling. So then the question came up to what happens to these neurodevelopmental defects here. I'm not going to go into all the details. The mice, both mice, either the N-terminal deletion or C-terminal deletion, they have narrower corpus callosum. That is one of the predominant defects, reduced size of the corpus callosum. They also have defects in internal capsule axon fiber tracts. This is magnified here. You can see the nice fiber tracts here, which you do not see in either of the knockout mice. So what do we see in the compound heterozygous mice is the corpus callosum defect is reduced, is rescued. So this is the narrower of the corpus callosum in 8 and 9 mice. This is the wild type mice, the corpus callosum. The compound heterozygous mice is indistinguishable from the wild type mice with respect to the corpus callosum defects. However, the axon fiber tracts and the internal capsule defects are not rescued by mTORC1 signaling, suggesting TORC signaling is not responsible for this. There has to be other targets for the axon fiber tracts defects and the internal capsule defects. Again, that's to highlight the defects that I showed you. So in summary, I've gone very quick. I didn't want to exceed my time. So what I have showed you is the impaired mTORC1 signaling in brains of these mice, either the Magellan mice with the C-terminal deletion or the 8 and 9 deletion at the N-terminus is seen in the brain but not in other tissues, and increased stability of the TSC proteins, which explains the mTORC1 regulation, is seen only in the Magellan mice, not in the N-terminal mice. And I've shown you in vitro and in vivo mTORC1 regulation. PAM regulates mTORC1 through functionally independent domains. And this may be partly responsible for the corpus callosum defects. However, as an E3 ligase, it may target other synaptic proteins, which may be responsible for the other defects, other synaptic defects that we are seeing in these mice, which remains important to identify other neuronal targets. That's something ongoing project in my lab. So what is the relevance to ASD? I'll finish with that. Dysregulation of MTORC1 signaling is characteristic of syndromic autism, including Fragile X and TSC. And also neurolegend, SHANK3, and neurexin, they all are connected to synaptogenesis. And it's becoming very clear abnormal synaptic homeostasis is probably a key risk factor for ASD. So at least I believe a key interplay between synaptic protein synthesis and synaptic protein degradation may be very critical in axon branching and synaptogenesis. And PAM could be an important player in regulating both synaptic protein synthesis and synaptic protein degradation. And identifying other neuronal targets of PAM may also be important as E3 ubiquitin ligase, what other synaptic proteins, including some probably ASD candidate genes, could be targeted by this interesting E3 ligase. And finally, I'm going to also mention, particularly, I was at the Simons meeting recently. They have a project called VIP Projects, phenotyping a subset of the ASD patients. Very clear by imaging studies, these patients have both corpus callosum defects as well as anterior commissure defect, raising the question whether this model, the PHR1 or the PAM mouse model, could serve as a valuable model for understanding ASD. The most important slide, people in my lab who have contributed in various ways for this project. Sang Yul Han started this project. He's back in South Korea, and several people are participating with ongoing results. I collaborated with Pradeep Bide to do some histology work, and the mouse models have been kindly provided by Aaron's lab and Sampaf's lab, and funding provided by Simons Foundation, NIH, and TS Alliance. Thank you.